So this is HER2 mutated non-small cell lung cancer. We have several trials ongoing with antibody-drug conjugate, so T-DXd, the DESTINY-Lung04 trial, waiting for the results. From the data generated in the late-line setting, this is a response rate around 50%. And a PFS about 1 year. So this is quite interesting strategy. But now we have also trials and data with HER2-directed TKI, so Zongertinib on one side and Sevabertinib on the other side. In the first-line setting, this is a higher response rate than with T-DXd, 70% response rate, and some duration of efficacy that is probably prolonged. We have seen the Zongertinib data during the meeting, median PFS 14 months, so quite prolonged PFS. But maybe the efficacy of the TKIs not only is higher in the first-line setting as compared to the later-line setting, and is also higher in the kinase domain mutations versus the non-kinase domain mutation. Still, the issue with one of the issues with the TKI is the safety profile with frequent diarrhea. 80% of the patients, more frequent in the late-line setting and after exposure to IO versus first-line, only 5% of grade 3 in the first-line setting. In the trials, the phase 1 studies, no systematic prophylaxis was implemented. The main treatment is loperamide to be started just after the first episode and avoid moving from grade 1 to grade 2 to grade 3. So probably the diarrhea is related to the exposure to the drug. At the end, it's not impacting the actual outcome and the overall survival. Still, this is something to educate the patients, clearly develop prophylactic measures, just to ensure that there will be no discontinuation of the drug. So during the meeting, I presented some of the data with Sevabertinib, but I think that these data are also applicable to Zongertinib. We are waiting for the results from the phase 3 studies in the first-line setting, and there will be many questions for these patients around the treatment sequencing between the TKIs and the antibody-drug conjugates.